Well, thank you, Chandan, for inviting me to give this talk. So to keep up with the informal nature of this presentation, let me spend a few seconds at the beginning with going over my background. So as you can see from this title slide that my undergrad was in civil engineering. And that was a very different world compared to what I'm doing now. But what I liked, one of the few courses that I liked in civil engineering was hydraulics, which is basically fluid mechanics. And then I applied for graduate school in fluid mechanics. I did my PhD from Virginia Tech in theoretical fluid mechanics, which I loved, the nature of the work, but I gradually realized that I need to train myself in other tools as well, beyond theory. And I took the first postdoc position in Japan, where the work was related to experimental fluid mechanics. And I soon realized that I'm not tailor-made for experiments. I don't like waking up in the morning and go to a lab and do the setups. So I wanted to move out of experiments. And then I took a second postdoc position at UNC Chapel Hill, where I had to first teach myself CFD. And I started working. So this was a position with the medical school. So I was involved in a number of projects where I collaborated with the physicians. We developed fluid mechanics models of or inhaled airflow, for example, in the nose, or maybe the airflow in the throat. We looked at drug delivery as well in those regions. So that's how, from civil engineering in the undergrads, I came to the world of medical science. The connecting link is fluid mechanics. So I'm using fluid mechanics tools to answer different types of interdisciplinary questions. So with that background, today I'm going to talk about a number of sub-projects that I have where I'm using fluid mechanics for different topics in medical science. More specifically, for this lecture today, we are going to look at problems in respiratory transport. So with this, let me go into the main material. So in terms of the overall theme of research in my group, I would like to classify it into two tracks. So first, I'm going to discuss about track one, which in my mind, it is more related to the translational application of fluid mechanics. I would come to track two at the end. Track two is more about exploring the fundamental fluid mechanics or the basic flow physics nuances in these problems. So let's go into track one first, which again is on translational applications. And therein, as you can see from these tiny little squares in there, I have a number of smaller projects. Today, we are going to just look at 2 or 3 of them. And I'm going to start with the first red one, which is on nasal drugs, or more specifically on topical nasal sprays. And before I go into that, again, I would repeat what Chandan said. If you have any questions, just feel free to stop me and then we can discuss at length. So the first project that I'm going to discuss is on nasal drugs. Now, irrespective of whichever project I might discuss, in general, when I'm using the fluid mechanics analysis tools in this kind of clinical domains, we typically always start from the medical imaging data. It can be CT scans, it can be MRI data. We use those scans to build models that are anatomically realistic. So as for example here, you can see this CT scan slice at the top right. So typically we collect around 250 to 300 such slices if we are trying to reconstruct the nasal airway of a person. And we get those scans and we digitally stack them up to build models that look like the ones that you see in the bottom. And once we have the geometry, then the next thing to think about is which particular question we want to address as part of some project. So for this step, I typically talk to the clinicians because they are the best people to know what are the open problems right now in medical science. So in this particular project, the clinical condition that we are looking at is chronic rhinosinusitis, or CRS. It's a very common condition. It involves some facial pain, loss of smell, a feeling of obstruction in the nose. And if you want to think of it in terms of the medical imaging, the 2 scans at the bottom, so the one on the left, it is from a person who is healthy, and those dark areas at the So at the center, those are the main nasal passage, and on the sides and at the top, they are the sinus cavities. They're attached like appendages to the main nasal passage. And you can see they're dark, implying that they're clear and there is not much mucus buildup. But if you go to the scan on the top right, you can see that those two big sacs on the two sides, they're the maxillary sinuses, by the way. And you can see that they have some congestion. There is some mucus buildup in those sinuses. And also there is some congestion in the main nasal passage as well. So that is a scan that we obtained from a person suffering from CRS. Now let's think of what are the possible treatments available for this condition. So in general, there are three possible avenues. So the first line of treatment are the topical nasal sprays. If that doesn't work, the doctors typically recommend orally administered corticosteroids. If that doesn't work, then the eventual option is to go for some corrective surgeries, which can help to ease the flow of air into the person's nose. Now, for this project here, we are not going to talk about the corticosteroids or the surgeries. We are going to think about the topical nasal sprays and try to figure out if there is a way to improve their therapeutic performance. So here we are just talking about the nasal sprays. Now, if we are just thinking about the nasal sprays, and we are also thinking about using tools from fluid mechanics to understand or to address this question on how to improve the spray performance. To keep the project realistic, or to keep the data that we generate realistic enough, we need to figure out what are the parameters in the currently available sprays that are out there. So for many of my projects, I collaborate with other groups with different kinds of expertise. So for this component of this particular project, I work with a drug development company called Next Breath. They're also based in the US. They're in Maryland. So what I did was purchase a few over-the-counter spray bottles from different makes, and I shipped them to them, this company called Next Breath, and they measured using laser diffraction techniques. They measured different parameters about the sprays. For example, what should be the size distribution of the droplets in the spray shot that is coming out, or what is the plume geometry of the spray shot, or what might be the initial speeds of the droplets of the particles that are coming out of the nozzle, those kind of things. And we can, once we get that information from this company, we can impose that into our simulations. So let's now think about the simulations that we have to handle. In this project, or in many of my projects, I tend to think of the simulation component in two layers. The first layer is, let's think about what is going on over here. We are applying sprays into the nose, and while doing so, the doctors typically recommend you to breathe in gently. So there is the ambient airflow that is being inhaled as well. So the first layer of simulation here is replicating that breathing process. And based on whatever flow rate we want to simulate, of course, you guys know that, or people who are from the fluid mechanics background, you know that it can be a laminar process or a turbulent process. Now, sometimes based on whichever CT scans we are using, if we had collected the breathing measurement data from that corresponding subject, then we try to replicate that particular breathing rate. Otherwise, there is enough data in literature where they correlate the weight of a particular individual, which we typically record whenever we recruit a new person into our study. So if we have the data on the weight of that person, there is some formulation out there, allometrically scaled, which helps us to figure out what should be the target breathing rate for that particular individual. So we simulate that breathing rate, and after that, the next step is tracking the particles. In this particular case, the spread particles from the nasal spray. We use discrete phase tracking, and the equation includes effects from the drag force that is induced by the surrounding airflow on these particles. There is the effect of gravity, and there is the Saffman lift force, which is generated by the flow field transverse to the direction of stream flow on these tiny particles. So eventually, the overall output from this component of the work are the regional delivery numbers in different regions of the nasal model, right? So we track where in the intranasal space, or which parts of the intranasal walls different particles are landing. So that is the overall output, but let's now think deeper into the question that we are trying to answer. is there a way to improve this targeted sprays? With this question, let's think a bit about the basic fluid mechanics here. So here I have shown the trajectories for two representative particles. On the left, we have the 5-micron particle. And if you look at it, that small tiny red circle is the point where the inertial motion of that 5-micron particle has stopped. And whatever it does beyond that point is because of the airflow streamline on which it was embedded at that point. On the other hand, if you look at the picture on the right, here I have tracked a 25-micron particle. It is somewhat heavier than a 5-micron particle, so it has a stronger inertial motion that will persist a bit longer, and eventually it gets deposited at the roof of the nasal passage where we have the ethmoid sinuses. So the moral of the story here is the inertia of the particles, what is the initial direction of the particles, that can play a huge role in figuring out where they might land as well. And there might be a possibility to use this kind of trend to improve targeted delivery of this kind of drug particles. So with this observation, let's think again about the question that we are trying to explore. And since we are trying to improve the performance, let's now figure out how these sprays are supposed to be used now. We are not changing anything about how the formulations in these sprays are designed or anything like that. We are just trying to figure out, is there another alternative way to use those sprays to improve their performance? And to answer that, we need to know how it is being used right now. So for this, you can look at the instructions that come with this over-the-counter sprays, or we can talk to the doctors as well. So typically, the patients are recommended to lean forward a little while applying the spray, and the spray bottle should be held vertically upright while doing so, and it should be just inserted into the nose. So these are the instructions that come with the package. Now, since we are trying to address this question through fluid mechanics, we have to find a schematic way to replicate this in the in silico world and run some simulations to figure out what might be the drug delivery for this particular usage protocol, which, by the way, I'm calling as current use, or CU. So the picture on the right shows you how I have interpreted these instructions. So I have inclined the digital head forward by an angle of 22.5 degrees. The spray bottle is held vertically upright, and it is inserted through a shallow depth of 5 millimeter into the intranasal space. And as I said, this is the CU protocol, and I can simulate how the sprays are going to deposit in different parts of the nasal model with this particular usage direction. So this is CU. Now the next part of the question is how to improve on that. To improve on that, we need to know a bit more about this particular clinical condition. So we are talking about CRS, and which is a kind of chronic sinus disease. And we talked to the doctors, as I showed at the very beginning from those scans, that in CRS, you have mucus congestion in the different sinus cavities. And we know that there are multiple types of sinus chambers. For example, over here, the sacs numbered 1, they are the biggest ones. They're the maxillary sinus. Then number 2 is, okay, so number 2 is something else. Number 3 is ethmoid sinus, which is sort of like at the roof of the nasal passage. 4 is the frontal sinus, which is right below the forehead. And there is, if you think about this, I don't think you can see my pointer on the screen, but the bulb-like cavity at the very end, that's called the sphenoid sinus, and I have not marked it here. But in general, there are 4 different kinds of sinuses. So in CRS, the mucus congestion would be there in these 4 types of chambers, but it is not possible to send our drug particulates to all these 4 different destinations, right? So we have to figure out a compromise. We have to figure out a common site that might be effective for all these different sinuses. So at that point, I talked to the clinicians, and they suggested that this region, which is marked by the blue circle, it is called the osteometal complex, or OMC. So OMC is the zone where all the sinus cavities, they drain their fluids into, and it is also the main airflow exchange corridor in the nasal passage. So if you can send more drugs to that region, chances are more drugs would percolate into the sinus chambers. So that is going to be our target site for improving the performance of nasal sprays. The next thing to figure out is, okay, so we have a target site now, that is the OMC. can we at all see it from outside of the nose? It is difficult to test this in a real person because then we have to obtain a number of permissions. So I 3D printed a nose. I made it out of a material which is similar to a real nose. So I have the same model here, if you can see. So it is soft like a real nasal tissue. And I used a speculum, and that blue chunk in there is the region where the OMC is. So indeed, it is possible to see the OMC, which is the target site, from outside. So now the idea is to use the inertia of the particles. To do that, I propose that let's orient the spray bottle such that the spray axis would cut through the OMC. And you can see that from the cartoon at the top right. And I call this as the line of sight protocol. I call it so because I can now directly see the OMC from outside. So the idea now is to compare between the CU protocol, which is already existing, and this new LOS protocol, a line of sight protocol. I tested the hypothesis that LOS would improve drug delivery on a trial subject first, and that is the data at the very bottom. I saw that the drug delivery increases by almost 8-folds in that particular individual. So that helps, that promises that this can be a good way to reorient the spray bottle, but we have to look at other subjects as well, right? So I included 4 other subjects. In total, in this study, I have 5 subjects, and in all of them, I see this improvement. The average improvement is still 8-fold, but the range of improvement is 2 to 16-folds, which is still very promising. But before we can be very sure about this improvement, we also have to validate the CFD model, because whatever we have done until now is in the computational world. So for that, I 3D printed some of the anatomic models, and I worked with another group. I don't do experiments, so for the experiments, I have to always collaborate with others. So what they did is take a nasal spray, spike it with a mildly radioactive element called technetium, and they would apply the spray into that 3D print, and they can then, once the spray solution they have deposited along the walls of the model, they can quantify the deposits through the radioactive signals that are coming out from those droplets that are on the walls. And the way I compare the CFD data with the experiments is like this. So if you look at the top row, I have virtually broken up the in silico model into these vertical compartments. So I first have the sagittal columns, and then the frontal columns, and then horizontal compartments, the sagittal rows. And I can extract the predicted drug delivery from the CFD from each of these compartments and compare that with the corresponding signals from the experimental data. I do this comparison. So again, I have the comparison for the sagittal columns, the sagittal rows, and the frontal columns. And on the horizontal axis for each of these plots, I have the different compartment ID numbers. And you can see that the deposition mass fractions are not exactly similar between the experiment, which is in yellow here, and the CFD data, which is in blue here. But still, the trend is sort of agreeable. And hence, I did a rank correlation test, and the coefficient comes out to be more than 0.9. So we can assume that there is good agreement between the CFD and the experiments here. So with this concludes the first project that I wanted to discuss. So what is the general outcome? So we have proposed a different way to use the nasal spray, which can be replicated very easily because the clinicians can directly recommend to the patients right during the clinic visit that this is how you should hold the spray bottle inside your nose, and that would give you a better drug delivery, which might increase the targeted delivery by almost 8-folds. So that is great. Secondly, this kind of findings can also help the drug development companies to improve the physical parameters of their solution to increase the targeted delivery based on whichever clinical condition that they're trying to address. So this is the first project that I wanted to discuss. Again, feel free to stop me if you have any questions. But if not, I'm going to go into some of the other projects. We are still on the track 1, which is on translational applications.
Saikat, I wanted to, I was curious that the 3D printed models that you use for the experiments, right? So were they based on some of your subjects that you tested?
Yes. So this particular example that I show here is one of the 5 subjects that went into this study.
I see. Okay.
Yeah.
And the other thing, like, could you please elaborate, like, how you actually estimated the correlation?
Okay,
like,
so
from the CFD and experiment.
Right. So for example, if you think about all these different compartments from the CFD, if you post-process the data, you can get the mass deposits from each of these zones, right? So suppose, let's imagine we are thinking about C11, C10, and C9. So let's imagine that we get X, Y, Z as the mass deposits in three of these compartments. I look at the corresponding compartments in the experimental data, and we see the trend. So if X, Y, Z, if suppose Y is greater than X and Z, if we see the similar kind of trend over here. So we do a rank order correlation in terms of, say, from smallest to the highest or highest to the smallest, and we try to check if that order is similar. Of course, they are not going to be exactly similar, but we found that the coefficient is more than 0.9.
I see. Yeah, we have another question from Manas. Manas, please feel free to unmute yourself and ask the question.
Okay, thank you very much. Your presentation was very interesting. I have 2 questions. The first being, what is the process you use to generate the computational model from the CT scan? Is it like a manual process where you use some sort of a software and do manually like smoothing or this kind of stuff to create a good enough geometry, or is it an AI or something based method to create the geometry? And the second question is, why is a steady state flow condition considered for the inhalation? These are 2 questions. Thank you.
Okay. So to answer your first question, no, we are not using any kind of AI here. So we get the scans in the DICOM format. We bring them into a software package called Mimics. I think there is another software that you can use called Slicer, but we typically use Mimics. And initially, we can, based on the radio density on those images, we can create some kind of threshold to get the bulk of the air space into the solid model. But then after that, my knowledge of anatomy is not that great, so I work with the clinicians in the team, and they help us in improving the anatomic realism of those geometries. So they would go through each of those 250 or 300 such slices and correct the pixels on those scans. And that's how we build the geometry. So we get feedback from the physicians that the geometries that we come up with are clinically realistic. So that answers your first question. What was the second question again?
Is the flow steady?
Yeah, right. So for the steady state assumption, the thing is, of course, if you are trying to simulate the breathing process over a certain duration of time, it is not a steady process, right? It is, first of all, it is periodic, and secondly, even if you just consider one half cycle, so just inhalation or exhalation, it is realistically unsteady in nature. However,
yes,
the kind of time domain that we are trying to simulate, especially if we are thinking about relaxed, steady breathing, in the sense that the person is sitting and breathing at a very low rate, that delta T amount of time, you can assume in all probability that the flow features are going to be steady because it's around 0.2 to 0.3 seconds at most. But of course, if you are trying to simulate on a wider time interval, it makes sense to, of course, steady would be a too stretched of an assumption in that case.
Okay. Thank you very much.
Okay, so next we go to Vikas. Please unmute yourself and you can ask the question.
I'm not having option to unmute myself, so I'm asking on the behalf of Prashant.
Yeah, okay. Yeah, please.
Okay. I want to know, can you suggest me some open source resources for this kind of data set, whether it is CT scan or MRI scan?
I don't have anything at the top of my head right now, but so typically we don't, for example, in terms of the imaging data, we don't use open source data. We are recruiting the subjects in a medical school, and we are building models for them, and we know their clinical history. But
Okay.
I know that there are some repositories out there. If you're interested, I can look it up, because I remember reviewing a paper a few months back, and there the group they were using some open access sources to gather the scans. The problem with those
Okay.
kind of sources is I had looked at them briefly a few months back for another project. The thing is, it's difficult to get the some of the information that you always need for realistic simulations. For example, what is the breathing rate, or even if you are trying to publish the data, what is the ethnicity of the person, or what is the gender? Those kind of information sometimes seem to be lacking. But I
Yes.
know that there are some online sources out there where you can get some scans. Yeah.
Yeah. And my second question is, have you considered the wall as a rigid wall, or are you considering the motion of the wall also?
No, this is a rigid wall. So that is one of the simplifying constraints that we have, and that is, and once I finish this talk, you will see that there are many open ends to this line of research, and right now we cannot really simulate the dynamic aspect of the breathing process, and these are all rigid walls. But these are great questions. So this area is going to be active for the next 10-20 years. So there is a huge scope to improve the modeling tools, one of which is not considering these walls to be rigid. There is another simplifying assumption here. We don't have so we know that in the nasal passage, there is the mucus coating, there is the ciliary hairs. Those kind of details are not there in this modeling framework yet. So there is a tremendous scope to improve this modeling platform.
Okay. Thank you so much.
Okay, so we'll take the question of Suman. Suman, please go ahead.
Hi, thank you. Thanks, Chandan. Thank you, Saikat, for the wonderful talk so far. Just a small question following up on what you mentioned a few moments back. The time scale of the problem being in the range of 0.1 to 0.6 seconds, is that something that you get from like the doctors or the physicians based on their experience? And what's usually the computational cost or computational effort for running the simulations?
Okay, so in terms of the time scale, see, if I look at the spray, the exit speed of these particles that are coming out, look at this number. It was 19.2 meters per second for that particular spray. Typically, the numbers tend to be always greater than 10 meters per second. So think about a particle that is coming out at that kind of speed, and think about the length that it has to traverse before it can reach, in this case, the target site, which is OMC. It's not that long of a distance. So the timescale was obtained as an estimate by us, not from the doctors. Typically, we know the timescales of breathing because on an average, I think there is a number for how many times we breathe in a minute. It should be around 15 or so, the entire cycle. So there would be like 15 complex cycles in a minute. So we can, of course, calculate those timescales. But here we are talking about how long these particles, these rock particles, are going to be airborne. And that is a very, very short time interval. So that answers the first question. So your second question, can you repeat that again?
What's the computational cost for your simulations? Like how much time does
for example, so that depends on the kind of numerical scheme that we are using. If it's a laminar, we don't use any kind of supercomputer, by the way. So we are using ANSYS, and we are using parallel processing on just workstations that we have in the lab. So for the laminar simulations, it takes around 4 or 5 hours. For turbulent simulations, we typically use LES. That can take around 3 days to 4 days. And if the problem is simple enough, and if we are confident that we can just use RANs, then that would take around 12, 13 hours.
Okay, thank you.
Yeah.
Okay, so we have another question from Manus.
Yeah.
Yeah, please go ahead. Yeah. Hello?
Yes, I can hear you. Yeah.
No, I'm asking Manus, do you want to ask the question, or is it the previous hand raised?
Sorry.
Okay.
I didn't know. Sorry, did I raise the hand again? I think it's a mistake.
Okay. Anyway, so Saikat, I have a follow-up question to Suman. So about the spray speed, that is something like clinically observed, and we do not have really control about. But then how do you control your, CFD's solver setup for the convergence? So from the, the time history, or I mean the flow output or flow field variables that we obtain from CFD simulation. So after, what is the time scale after which they get converged normally? This is just.
You mean the time scale in the simulation, right? So
yeah, so since you are considering steady state, so like after how many time step or iterations, like normally?
So if you think about, so let's think about the steady state. Let's think about the laminar simulation. So I'll give you some sample examples. So if we are trying out 5,000 iterations, we typically see stabilization from around, I would say, 1,000 or 2,000 iterations on an average. And if we see that the residuals have stabilized, we might not even go till 5,000. But that is a typical number, yeah. And that can vary from one subject to another. Yeah.
So what is the minimum time step that you have gone for? Yet, I mean, just for a number, let's say.
The minimum time step. So, for example, it is around, for the earlier simulations right now, based on how what delta t we are trying to simulate, I have a time step of around 0.0001 second.
10 minutes. Okay. Thank you. Yeah. There is no question, no more question at this stage. Please go ahead. Yeah.
Okay. Okay, so the other projects are going to be relatively smaller compared to the discussion in the previous one, because I wanted to cover more details on that one to give you an overall flavor of the modeling framework. Now, this one is for nasal surgeries. Fluid mechanics can also help us figure out what would be a good surgery for a particular individual. And here I am again looking at a particular clinical condition called nasal valve collapse, where the opening to the nose gets constricted. And there are three main surgical corrective procedures for this. So these are the cartoons that we have here. The first one on the left is the butterfly graft, where the implant is placed transverse to the direction of the flow. Then I have the spreader graft, where the implant is longitudinal. And then the third one is the lateral graft, where it is similar to spreader, but here the implant is bioabsorbable on the very right. Now, the spreader grafts have been around for a while. Butterfly graft, this technique was developed over the last 20 years or so. And lateral graft, the one on the very right, it's really new. It's around 2-3 years old. So the question is, to correct the anatomy for nasal valve collapse, which one would be the best procedure? And here again, I worked with the clinicians. What they did is collect a number of cadaveric heads. They carried out this surgery, all three types of surgery in each of those heads. And they collected scans both pre-surgery and post-surgery. And we used the scans to build our geometries, and then we ran some airflow simulations. The main quantity that we post-processed from the data is the airflow resistance, which can be computed as the pressure drop that is driving the flow divided by the flow rate. And we see that with the butterfly graft, post-surgery, the improvement in the airflow resistance is almost 34%. And this is from a cohort of around 15 cadaveric heads. The spreader graft and the lateral graft, their performance is very similar, but lateral did somewhat better. So at least for these 15 subjects, we can tell that butterfly graft works more efficiently in terms of helping the flow of air through the nose compared to the other two techniques. But what is the general moral here? The general storyline here is this. We can use CFD, we can use fluid mechanics in a number of different ways. As for example here, if a clinician has recommended medical scans or CT scans to a particular individual, we can use our modeling framework to develop a full visual of the airflow patterns, the drug delivery trends, the outcomes for different nasal surgeries by using virtual surgeries into this in silico geometries. And we can have that data available to the clinician, which can help in the follow-up diagnosis, or it can help in figuring out what could be the best type of drug for that particular individual, or what could be the best surgical process for that individual. So this can be the next layer of how engineering can mesh well with medical science. And this would create a platform for more personalized healthcare. So that is one topic that I wanted to cover. Let's now move into something which is somewhat different. And this line of my research essentially took off over the last 1 year, of course, with the outbreak of COVID. But as you can see from my projects, I've been working in this kind of nasal transport process for the last 5-6 years. So COVID-19 being primarily a nasal disease, it helps to have that background and answer some questions that we are facing right now. And as of now, I have collaborations related to COVID-19 research with a number of clinicians and a number of pharmaceutical companies. And we are also involved in drug development. Well, by drug development, I mean some therapeutics that can help in relieving the condition. We are also working on intranasal vaccines. But for the discussion here, I'm going to talk about the project that I started on my own last year to figure out what we know that SARS-CoV-2 is an airborne transmission, right? So the question that I started out from, and this is, I would say, around April of last year. Okay, so if it is an airborne transmission, which are the bad droplet sizes? We know that in the air, the droplet sizes that we eject through breathing or talking can be anywhere between 0.1 microns to 500 microns. But what are the bad sizes over here? To answer that question, I had to again fall back on the literature that was coming out at that point, and it is still coming out. And there have been a number of studies which looked at how this infection starts. So I have just cited one example here. This came out on Cell. They looked at how the infection progresses, and they saw that the infection starts in the ciliated epithelial cells, which are more abundant on the upper airway. And these cells, they have a certain kind of protein called ACE2, which the virus uses to intrude into the cells. So these cells are more abundant, as I said, in the upper airway. But if you think about the upper airway, there is also this conjecture that there is a thick layer of mucus in the anterior cavity. So a number of groups, and that includes me as well, we have posited that the initial dominant infection site for SARS-CoV-2 is this red zone over here called nasopharynx, because in the anterior parts there is too much of mucus which provides some kind of preventive layer or protective layer against the virus invasion. So the infection is dominantly starting out from the nasopharynx. So if that is the case, can we figure out the droplet sizes that will directly land at the nasopharynx through the process of inhalation? So with that overall hypothesis, and to answer this question, let's now go into the actual work that I did. I ran a number of inhalation simulations, and I varied the airflow rate from 15 to 85 liters per minute. Now 15 liters per minute is something that we are breathing in when we are just sitting steady. 85 liters per minute is too high of a spectrum. It is typically what we would be breathing when we are running or breathing in forcefully. And I chose these particular numbers because these are also the numbers that biomedical device companies use to test the filtration on respirators or face coverings or masks. So I ran the breathing simulations through all these different inhalation rates, and then I ran some particle transport simulations as well, or droplet transport simulations. And I see that for targeted landing at the nasopharynx, the typical size range for those particles, if I just look at the average data over the entire spectrum of different breathing rates, that size range is from 2.5 to 19 microns. And one critical thing to note here is, here the density of these droplets is 1.3 gram per ml. Why? Because these are the droplets that are ejected from someone who already has that infection, right? So it's made from respiratory ejecta. And we know that those respiratory droplets are formed from sputum. Sputum has 99.5% water. But once those droplets come out into the air, they undergo environmental dehydration. So out of that 99.5% water, much of the water would vaporize off. What would remain are the other remnants which are non-volatile, and that includes the virus as well. So the density would increase appreciably to around 1.3 gram per ml, which is again an average number that comes from other papers. So for this particular density, we see the size range, the batch size range is 2.5 to 19 microns for delivery at the nasopharynx. One may ask the question, what if the geographic region that we are thinking about it has a high humidity. So what if it is, for example, India? So if the humidity is high, the environmental dehydration would be low, or yeah, it would be very low. And what would happen as a result, that sputum, which was 99.5% water initially, the water won't be vaporized off. And as such, the droplets that the exposed individual is going to inhale would be similar to water droplets. And over here in these simulations, I hence maintain a density of 1 gram per ml. And even with that, I see that the hazardous size range does not change much. So from 2.5 to 19 microns, it gets revised to 3 to 20 microns. So the droplets are still of the of somewhat similar sizes. So with this, I think we have answered the question that we started out from. So what are the bad droplet sizes? The bad droplet sizes are somewhere in between 2 to 20 microns, or 3 to 20 microns. And that can actually help us in many different ways. That can help us to come up with better designs for new kinds of face coverings that will just screen these bad particulates out. And hence, in the process, they can be more breathable than the N95 masks that we have, which actually try to screen even particles which are as small as 0.1 or 0.2 microns. So that answers the first question. The second question that I was exploring for the last few months is, what is the infectious dose? Some of you might know what is an infectious dose. Infectious dose for a viral condition is defined like this. It is a number of virions that can go on to start an infection. So we know these numbers for the different flu viruses, but this is still a very important open question that we have for SARS-CoV-2. We don't know how many virus particles or how many virions we need to inhale to get sick. So to address that question, I was again looking at literature and the papers that were coming out. This is a very important article that came out on PNAS. Here they tried to figure out what is the probability that a droplet of a certain size would carry at least one virion. So for example, they found that for a 10 micron droplet, if it is undehydrated, so there has not been any environmental vaporization yet, then the chances that that 10 micron droplet would have at least one virion is just 0.37%. If on the other hand, the size is 3 microns, the probability to carry at least one virion is around 0.01%. So I decided to develop a simple mathematical framework to test these numbers. And in the process of developing that framework, I again had to read some more papers on virological assessments. And this is one paper that came out on Nature last year. So this is a German group. They collected virological data from a group of hospitalized COVID-19 patients. They figured out the RNA viral load in their sputum. And this SARS-CoV-2 is a single-stranded RNA virus. So we can assume that 1 milliliter of oral ejecta would have that many number of virions, 7 times 10 to the power of 6. And hence, a droplet that is being formed from that ejecta, if it is of size d micron, then this capital N would be the number of virions embedded in that droplet. In other words, to come across one virion in these droplets of d micron size, we have to test n inverse number of droplets. And we can put this in terms of probability as 1 over small n percentage. So this 1 over small n, or the p function over here, is a function of the size of the droplet d. If we plug in the value of d into that equation, we can see that p is 0.01% for 3 micron size and 0.37% for 10 micron size, which matches almost exactly with the numbers that were reported in the other article. So with this modeling framework, I'm going to use this third equation where we calculated how many virions are there in a droplet of a certain size, and couple that with the CFD data that we have on which are the bad droplet sizes, which are the sizes that are depositing at the nasopharynx. So I link these two data and I figure out that if you are exposed, if I am exposed to an individual with an average viral load, I am going to inhale around 11 virions that are going to land directly at the nasopharynx over a duration of 5 minutes. But on the other hand, if the infected individual has peak viral load, I can inhale as many as 3,800 virions over 5 minutes that are going to directly deposit at the nasopharynx. So with this data, we can look at other super spreading incidents that have been reported over the last 1 year to figure out what was the exposure time during those incidents. And I can come up with a number which might lead to the start of this infection. So the exact number that I calculated in my work is around 300. And right now there are other papers that are coming out as well. Most of them are preprints at this point. They are projecting similar numbers as well. For example, one of these papers, they said their number is 280. So which is great, because we typically just want to come up with the order of magnitude estimate. And to put this in perspective of what about the other viruses? So for example, for influenza A virus, the number of virions you need to inhale to get sick is around 2,000 to 3,000. So this shows that SARS-CoV-2 is almost 10 times more contagious than a flu A virus, because the order of the infectious disease is just one order smaller. Okay, so that I think wraps up the projects I wanted to discuss in terms of the translational applications. I can pause here for a moment. I have around 10 minutes of talking time left in terms of the material I want to cover. If you have any questions, you can ask me now, or I can move on.
So yeah, so do you guys have any questions? Please feel free to ask.
Okay, so let me just wrap up the talk and then we can discuss a bit more.
Sure.
So on the track 2, I think about the basic fluid mechanics that might be impacting this kind of transport processes. So for example, let's think about the vortex dynamics. We know that vortices, they have a low pressure at their centers, and it is possible for them to trap the drug particulates, which can mess up targeted drug delivery if the target site is downstream of that region where the vortices have emerged. And people have looked at this kind of problems, but in most of the cases, the geometries that folks from the mechanics community have used are idealized. For example, like a simple T section over here. What I'm trying to do is to think about the anatomically realistic geometries and explore how the vortices might impact drug delivery. So the particular problem that I'm going to discuss here is related to a condition called vocal fold granuloma, where there are these little tumors that are formed near the vocal folds in the throat. I have virtually implanted tumors of different sizes, and these sizes are decided based on my discussion with the clinicians. And then I have run the breathing simulation and then some particle simulations to figure out what are the ideal particulate sizes for targeted delivery around the vocal fold granulomas. And I see that the size range is between 8 and 10 microns. Now let's compare that to what we have right now in terms of prescribed drugs. There the size range is 1 to 4 microns, quite different than what we see from our modeling work. So this shows you that it is indeed possible to use this kind of fluid mechanics tools to even improve the design of these targeted drugs. And hence I have a number of collaborations with the drug development companies as well, who want to explore these kind of nuances and use fluid mechanics in their working process. The last project that I wanted to discuss is about bio-inspired mask filters. Here, this is a project that we recently got funding from NSF for. And here we are trying to come up with a design for a novel kind of face covering, a filter. And what we are thinking about is this. We know that in the animal world, there are some high olfactory animals, which they have a very complex nasal passage. As a result, when they inhale, they can screen the particulates much more efficiently from that inhaled air, and that helps their sense of smell. Can we use that kind of design in terms of structuring the filter pathway, which will help in trapping the particles, and yet, because it is bio-inspired, the pressure drop for the flow through that filter is not going to be that high, because otherwise that is not going to be supported by evolution of the animals, right? And while doing so, we are still going to capture the particles that are actually the bad particles for disease transmission. And again, the project that I just discussed, what are the bad droplet sizes, can help in figuring out what should be the design of the filter, because we just want to trap those particular particles out. And in terms of the pressure drop, that is a measure of the breathability of the mask. Our main goal is to make sure that the pressure drop through these filters is lower than what we have, for example, from the N95 masks. And that would imply in qualitative terms that the mask is going to be more breathable than the most advanced face coverings that are out there. So this is a sample design of the filter pathway. We used a tortuosity value of 1.9. On the right, you have a single pathway, and around 300 of them would go into a circular domain to make the entire filter. So this is a collaborative project. There are other groups who run the experiments. So for example, the pure flow experiment is being run at Cornell, where they measured the pressure drop across the filter. And at UIUC, they're simulating, they're testing experimentally the filtration performance through these filters. The work that I'm going to show here is basically replicating the experiments that they're running at Cornell University. So on the top left, you have their experimental setup. I developed a similar kind of in silico domain that we can run CFD simulations for, and here we show the pressure drop data for different kind of breathing rates. As you can see, the pressure drop range varies from very small to around 250 pascals, but still, if you compare them to N95 data, the pressure values are smaller, which in other words imply that these masks are going to be more breathable than the N95s. And the other layer of this project is to filter the particles. So this is the heat map for particle filtration. And we see that the design that we have right now, it can filter the bad particles out most of the time. And this is a sample video for to show you guys where the particles are getting trapped in a single filter pathway. So at the very beginning, the particles are being trapped because of the inertial motion. And as we go further, the trapping happens because of the fluid mechanics. These are for 3.5 micron particles. So if you think about the particles that are getting trapped further downstream, they are being trapped thanks to the vortices that are emerging on the system. And they're being sucked towards the low pressure zones on the walls. And this screenshot tells you the overall trapping trends for different particle sizes. You can see that at least this particular filter design did not do a very great job of screening the 2 micron particles. They did a sort of good job with the 3.5 micron particles. And for 10 micron particles, almost all of them were trapped at the very first bend of the design. And over here, I show some videos to show you the general trend of the particle dynamics. So based on the initial location of the particles at the inlet of the filter, they would be trapped or be able to escape. So on the top row, I have 3.5 micron particles. At the bottom, I have 10 micron particles. So the 3.5 micron particle got trapped, the one on the top right, but both the 10 micron particles are getting trapped. So with this, let's wrap up. So as you can tell from the overall flavor of my research, that yes, I'm bringing in the fluid mechanics expertise to the table, but to answer these questions, I need inputs, I need expertise from other groups as well. So I work with a number of medical schools. I work with a number of engineering departments, both mechanical engineering and biomedical engineering. I have a number of collaborations with the pharmaceutical industry. And on the right, these are my funding sources. I'd like to end with this slide. These are some words from Schrödinger. So he said in this book, What is Life, back in 1944, that we are only now beginning to acquire reliable material for welding together the sum total of all that is known into a whole. So let's think, this is 1944, World War II is going on, and we are now in 2021. So as engineers, or maybe in this group, many of us are engineers, many of us might be physicists or applied mathematicians, we have a lot of sophisticated tools which we can use in many different ways. So for my group, I'm trying to use the tools that I have learned through my academic career to answer these open questions in medical science. So with this, let's end this talk, and I'd be happy to discuss more.
Okay, thank you so much, Saikat. So we are now open to questions. So formally, so please, feel free to raise your hand or stop us with your questions. So before anybody asks, I have a question. So it's a kind of broad curiosity question. So have you started characterizing the vortices? Like we are telling that some underlying vorticity dynamics is responsible for the trapping, right? So
to be frank with you, no, we have not yet. But that's a very good question, and which I want to explore as I move along. So the challenge over there is, I think you know some of my earlier work on Vortex Dynamics, where we developed 2D potential flow models for vortex tracks, but that is too idealized of a system to apply in a real world. So these are all 3D flows. So that is going to be a challenging line of research, but something that I want to explore moving ahead.
Okay, it will be really interesting.
Yeah.
Okay, so does anybody have any other question?
I can ask something, but this will not be that much
Please go ahead. It's just an informal session.
Saikat, it's Shuman here again. So this is coming from fluid mechanics, and I think Chandan also fluid mechanics. I think more closer to what your work was in Virginia Tech, because he also works on vortex dynamics.
Right.
I'm mechanics, but more at a molecular level, and that's why I was asking you the timescale questions before, because my simulations are more on the level of nanometer and nanoscale timescales and length scale.
Let's see. Yeah.
So my questions would be coming from like Dr. Stremler's group, which is like more theoretical vortex dynamics. Like, at what point did you feel that you want to shift to a more application-oriented research? Was it like just after the PhD, you wanted to start searching for application-oriented domain for research to expand your research, let's say, domain or expertise? And the reason I ask this because like I'm like planning to wrap up my PhD in a year or so. Started looking for postdocs, and coming from a molecular dynamics perspective in fluids, and the way US research plays is like it's a very application-oriented domain, right? So it's very difficult for theoretical people or let's say pure science people to survive without the way it is. So at what point did you feel that you want to shift to application or more you explored experimental as well, and what made you choose medical applications to be the field that you want to explore first, and maybe you can diversify later as well?
Okay, yeah, that's a very great question. First of all, but before I go into my answer, you are at Purdue right now, right?
I'm at Purdue, yeah.
Yes. Yeah.
Who are you working with?
I'm in the aero department. I'm working with Lee Chow. We are a combustion lab. She's an experimental person. I'm the only computational person in the group. So she mostly works on sprays on nanoscale combustions, but I mostly study phase change and transport properties at molecular level.
Great. Okay. Anyway, so yeah, that's a great question. So let me try to answer that. So when I was working at Virginia Tech, I was very happy working in those kind of vortex dynamics domains. It was intellectually very challenging, and I enjoyed my dissertation. But as I was wrapping up, that should be around end of 2013, and then I graduated in May 2014. While I was wrapping up, I saw from my seniors at that time that I wanted to be a faculty for sure. I did not want to go into industry R&D. So I wanted to be a tenure track faculty, but I saw that if you're just from a theoretical research area, it is difficult to land a tenure track position. So that was my first cue that I need to know a few other things beyond theoretical fluid mechanics. Folks could have landed a tenure track position 20 years back with just a theoretical background, and we know people like them. But I don't think that is possible anymore. You need to diversify your tool sets. At least that was true for me. So what I was doing was too theoretical in nature. And point vortex dynamics is interesting, but it can only be applied to so many real life problems, right? So the next thing that I did was I thought that maybe I should learn some experiments, because at that time I was still a graduate student. You come under some kind of notions that, oh, it might be easier to run experiments and whatnot, which is not at all true. So I thought that, okay, let's do some experimental fluid mechanics. I was applying for postdoc positions. I started quite early. So I was about to graduate in May 2014. I think I started looking for postdocs at least 10 months before that. Eventually, I got lucky. So I was interviewed first on Skype by Mahesh Bandi at OIST, Okinawa Institute of Science and Technology. So I knew Mahesh because he visited us at Virginia Tech as well before. So he interviewed me there and through Skype, and then they flew me over to Japan where I gave my research talk, and they were an experimental group. So I thought that, okay, let's run some experiments, because the project that he proposed for me was related to Vortex Dynamics. He proposed that I would have to design a vertical soap film setup where we can run some VIV experiments. That was all fine, but the moment I started doing experiments, I realized that I'm not really enjoying myself over there. But even then, that process helped. So I was there for around a year and a half, and I learned more about the experiments. And hence now when I work with other groups, I'm not under any kind of notion that experiments are easy. They're difficult, and we need to know more about experiments, even if you are a computational guy or even if you're a theoretical guy. So that stint helped. But to go back to your question, at what point did I want to diversify? It was the point when I realized that just having one single strength is not sufficient to land a tenure track position. You need to diversify yourself further. But since I did not enjoy experiments, I knew that I need to know something else as well. So that's why I had to go for the second postdoc. So I was applying for different positions, and eventually I landed this postdoc position at UNC Chapel Hill. It was on computations because, of course, as a fluid mechanician, I'm done with theory, I'm done with experiments, so that's the only other option left. So I wanted to do computations for sure. But I did not know what would be my application area. So that was a matter of luck, I would say. I knew that medical science has a lot of open problems, and we all know that biomedical sciences, they are going to thrive over the next 20, 30 years. But I did not have any specific decision as such that I'm just going to work in medical problems at that point. And this is 2015. So end of 2015. But I landed that position and I started working with the clinicians. The initial few months were rough because I had to know a lot of things about computations which I have not done before, or I had to know a lot of things about the medical anatomy, those kind of things. But it was a good experience because finally I realized that whatever esoteric problem that I was working on in graduate school, and for that matter even in Japan, I can now use those tools in something that can directly impact the world around me. That, at least for me, was a great feeling. Like now I can actually apply my fluid mechanics knowledge in a real life problem. And so I kept on working, and then I started developing projects of my own. And to be frank with you, there are not many people like me in this area where people are working in this kind of respiratory flow problems with a fundamental background in fluid mechanics. So that gives me some edge. So I would say for people like you who might want to become a faculty later on, try to find that niche area where your strengths might come in and be very effective, while at the same time do make sure that you build up on at least 2 or 3 skill sets which you can keep on applying to different problems as you move along. Because as a tenure track faculty applicant, you need to present a cohesive story for your application package, a cohesive story for your research program, and all your skills should come in together to design that program for the next 3 to 5 years.
Thank you so much. Yeah, that does. It's good to get a perspective from someone else who has actually walked the path and is actually where we want to end up at someday, hopefully.
And let me tell you, getting a tenure track position is very difficult. I did not realize that back then. It's really difficult. So be prepared to send out a lot of applications.
Yes. And on a same note, or let's say on the same line, how much open do you feel professors are to accept someone who's from a theoretical or a computational background, to allow them to explore something new in a postdoctoral time?
I think that is subjective. It depends on the particular PI. Because I mean, postdoc should be ideally used to build up a new skill. I know many PIs who just try to intake someone who already has that skill. Somehow I was lucky, so I got the chance to build up on new skills. So that is something that you need to consider when you decide on your postdoc position.
Okay, thank you so much. I'll let others ask questions. Thank you so much, Saikat.
Yeah. Chandan, you are muted.
Yeah, so thanks, Suman, for asking, and this discussion was very enlightening actually. So anybody wants to ask any other question related to the talk or outside it? Okay, Manas, do you want to ask a question?
Yes, thank you very much. It was really a wonderful presentation. I have one question regarding the geometries that you prepare. So what software you use for generation of the mesh?
The mesh is being generated on ISM CFD.
ISM CFD. Okay. Okay, thank you very much.
Okay, so I have a follow-up question. Like, I mean, I think in CFD community, in this kind of nasal airflow problems, the slicer is very, I would say, very popularly used. So, any particular reason that you are not using it?
Not really. So I have a collaborator at Duke. He's using Slicer, and yeah, I mean that it works as well. I have just not used it. Like I recently have a new graduate student who is trying to learn Slicer. In terms of the modeling framework, it's similar to what we have on Mimics. It's exactly similar. You have like a coronal view, an axial view, and a sagittal view, and you try to build the model.
Okay. Yeah. Thank you. Okay, so if we do not have any more questions, I think, yeah, let's thank
one last experience, Chandan, that I would like to share, going back to Shuman's question.
Yeah, sure.
When I was being interviewed during that tenure track cycle, I still remember, I'm not going to name the university. It's one of the top 10 R1 universities where I was invited to for the on-campus visit. It's in the south. There, the department head, after the interviews, he told me directly that the papers that you have written, the ones that you wrote from Vortex Dynamics, they will stand probably the test of time. They are the best in quality. But we did not shortlist you, or we did not invite you here because of that work. We invited you here because of your work in biomedical sciences. There is a higher chance of getting funding. So this is the reality. So you need to be prepared for that. So make sure that the program that you propose should match your own interest, because you cannot just think about funding. It should be of your own interest as well. But still, you need to make it packageable enough to what the funding venues would like, or what your department chair would like. So that's just the reality right now out there.
Yeah, because that's what the research market is going through right now. It's just I feel like at least in more theoretical and basic sciences, it's much easier to find, let's say, like-minded people over in Germany and in Europe who are still happy doing theoretical basic research. But US is such a fast-changing and dynamical research market that I guess it needs to change with time. Otherwise, it's difficult to survive.
Yeah, I mean, personally, I don't complain because I like the work that I'm doing. But
oh no, definitely.
If you ask me, like, if you ask a 2013 version of me, I would have been very scared, because whatever I knew back then was just point Vortex Dynamics. But it is important that you really discover yourself as you go along. Yes.
Yeah. Thank you so much for the advices.
Yeah.
Okay, Prashant, do you want to ask a question?
Yes, sir. I want to remind Professor Saikat to suggest some open sources for the MRI scans for the upper airway tract as well as the lungs.
Okay, yeah, I will do that. I probably don't have your email address, but I can send. If I find something, I'll send a link to Chandan.
Okay.
Please share your email ID in this chat, or in my. You can find my email ID from the website of this talk. Okay?
Sure. Thanks.
Okay. As I said, I did review a paper which I think came out from Singapore, and I can look it up, and if I find something, I'll let you know.
Yeah, sure. Thanks.
Chandan, I'd also be very much interested. Chandan, can you hear me?
Yeah, please.
Yeah, I'd also be very much interested in that question regarding the open source
data.
Yeah, definitely. So kindly send me that.
Okay.
Thank you.
Yeah. If Saikat sends me, I will share with you guys. And I think you both know my email ID. Just share your email IDs. I will.
Okay.
Thank you, Saikat. It was a really good talk. Although, like, I have I know your research, but for the others, I think it was very interesting, and hope to see more on the interesting vortex dynamics from your work.
Thank you for inviting me again.
Yeah, and thanks everyone for joining the talk, and it's really a pleasure to have you all here. So I'm closing this session. So probably we'll share the next talks details in the WhatsApp group or whatever social media platforms I use. And thank you everyone once again. So I'm closing the talk here. Bye.